Galaxy training tutorial, exome sequencing data analysis for diagnosing a genetic disease. It's a really long title to remember, but yeah. Estimated time is about 5 hours. And yeah, from my experience, you're gonna need that type of time, not necessarily to do the work, but just because how long it takes for the data to load and process on the cloud. Okay, step one. Okay, let's actually just scroll down. Introduction. We're introduced to a family. Where is it? Yes, here. A family where there's a boy with osteoporosis, but both his parents don't show any signs of osteoporosis. So we're going to be doing whole exome sequence analysis, more or less, and taking a look at what might have gone wrong. First step is to create a new history already created, and rename the history something you're going to remember. So in this case, I'm going to say exome analysis and then dash osteo. I'll leave it there. Yeah. So next, starting with raw sequencing data, we can more or less copy this information directly. And then you go to upload data, paste, and then you start the process. That will load onto Galaxy. Give it a couple minutes, should be fine. Okay, next, assuming everything ran the way it should, all the files should show green, and it wants us to confirm, or Galaxy wants us to confirm, that we have the right database types. So FastQC sanger.gz file, and just go through all your files, make sure they're all more or less in this format. If so, then you're good, everything should be fine. And it says, congratulations, proceed to step 7. The next few steps are if you didn't load the data using that method. Okay, so the remaining steps, rename the datasets. You obviously want to make names that are easy to remember. It's already kind of given to us, the father, mother, proband, so I use those, and I just remove the extension names at the end. So the .fq.gz, just get rid of that. Save, and repeat the process for the rest of your files. Okay, now that that's done, we can move on to the next step, but please, for your sake, do keep the R1, R2 at the end of each of the names for each data set. It does make it a lot easier to work with the data later on. You don't have to, but trust me, it makes things much easier and less confusing later on. The next step we have after renaming things is to add the tags like father, mother, and this is really helpful later on. You'll understand as you go through it that, okay, you can see I already have the tags there because I've done this data set before. This is my second attempt just to show you guys what's up. But as I was saying before that appeared, it really is important that you tag everything, because as you move from data sets and you analyze them, filter, blah, blah, the original names, father R1, mother R2, proband R this, R that, whatever, disappears, but the tags do stick with each data set. And the tags are exactly what it is, the tag that follows through the entire process. So you don't mix up the data and you know exactly what's going on. So let's go ahead and tag the data. So you can just copy directly from the add tag, paste father, add tag father, next mother, finally proband, so in this case it'd be child, add tag child, add tag child, and yeah, That simple. We now have father R1, father R2, tagged father, mother R1, R2, mother proban 1 to obtain the reference genome. You can skip this step if the Galaxy server you're running has the HG19 genome. Fortunately, we're using the EU server, and it does, as well as the .org server, and I'm not sure which other variations of Galaxy has them, but those are the two I've worked with. And a heads up, if you do plan on changing servers, you do need to recreate an account. For some reason, they don't recognize that each of the accounts. Really weird fact to know. But yeah. Okay, once that's been obtained and stuff, you can more or less move on to the next step. Congratulations, you're now set for analysis. Okay, so the next step in this analysis would be quality control. It's more or less there to make sure the quality of the input data we're getting is good enough to move forward and actually get an analysis done. You can also work through here. Run FastQC on each of your 6 data FastQ datasets. Short read from current history, all 6 of FastQ datasets selected with multiple datasets. So that's what you're gonna do. See where it says run, it says the short read data from all your current history. Multiple datasets, you can do that. Select, 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 and select. So 1, 2, 3, 4, 5, 6 is all selected. And you don't need to add anything else. Selectable mode, multiple datasets. They tell you need to hold command or control. And when you start the job, 12 new datasets. One calculates with raw data, HTML reports, and etc. will get added to your history. So that's all we need to do. Run tool. And this can take some time to load. As you can see, 7 through to 18, which is 12 datasets. It's gonna take a minute to load. So now that the FastQC step is complete, we do a multi-QC. It's more or less the same thing. It's just to summarize all the data together. So yeah, multi-QC tool is here, and it tells you use multi-QC to aggregate the raw QC data in results, which tool was generated, which tool is used to generate the logs is FastQC. So you can either type it in or you can search it there, FastQC. In FastQC output, type raw data. Okay, where is that? Insert FastQC output, raw data, there we go. And FastQC outputs is all 6 raw data outputs of the FastQC. Okay, so it sounds complicated, but it's not. Multiple data sets, it's already selected for you. Open up raw data. There should be 6 files. You can compare them side to side. 18, 16, 14. That's all the raw data files. And this should more or less aggregate everything into one giant multi-QC file. Okay, that shouldn't take too long to load. Should only take like a minute or two, and once that's done, we'll move on to the next step where it says inspect web page output produced by the tool. Okay, so this is really important to get an idea of what's going on with the data, what could be improved, and whatnot. Just looking at data like this, you don't really see anything out of place except for the per base content. You can see it could be a little better. 6 out of 6 samples have warnings on them. Take note of that. And over here, the GC content, you can see it's a red flag. 6 out of 6 samples failed. So the GC content's really high. Per base N is normal. Everything else seems normal. Green, green, green. So what does that tell us? It tells us that the per base sequence contents could be improved and the GC contents could be improved. Processing, blah, blah. And then, as we pointed out, yes, it could be improved, and here's a solution. For the solution, they say sequence quality is quite good overall. If anything, you might consider trimming the 3' ends of the reads. Base quality declined slightly toward the 3' ends, or to filter out small fraction of reads with the mean base quality below 5. But I don't expect this to have a big effect on the analysis given the high overall quality of the data. In whole genome sequencing, a non-normal distribution of GC content of the reads from a sample is typically considered to hint as possible contamination. Here, however, we're dealing with sequencing data from captured exomes. That's something you should take note of. We're running exomes, not whole genomes. So it has a whole different meaning to it. I.e., the reads are not representing random sequencing from a genome, but rather a bias selection. That's the difference you get with exome sequencing versus genome, which is why we don't worry too much about the GC content. Like, yes, it's not the best, but it's a very biased sample in the first place. So we do expect there to be some deviation from norm. A bimodal GC content distribution, like for the samples at hand, is a characteristic feature of many exome capture methods, and also has been observed with Illumina blah blah exome kit before. Okay, for some reason the Trimomatic tool is not working for me today, so we're skipping that step, I guess. As stated, it's not really a necessary step, it's just nice to improve the data. Okay, next step is read mapping. Now that you confirm the quality data is good enough, even they said good enough, we can carry on further analysis. So assume here you're at least vaguely familiar with the concept of read mapping and only illustrate the concrete steps necessary to map next generation sequence reads datasets to human reference genome. Okay, so we can click on tool number 1. We'll be selecting reference genome from history or built-in. In this case, it should be built-in. And then reference genome is HG19. This is something you have to take note of. Be very careful with. We're using HG19, not the 38 and not the 37. They may be considered more updated versions of this, but from comments you can see at the bottom feedback and stuff like that. It doesn't always work, so rather just stick to the 19. We know it works. Single paired or paired ends. We're using paired ends for more accuracy. And then select first set of reads. This would be R1 for the father, and then select second set of reads. That'll be R2 for the father. Okay, so in this case, R1 for the father, R2 for the father. Select first set, second set. Not that hard to keep up with. Next, after doing that, is set read groups information. Okay, set read groups information. The SnpEff specification is what we want over here, as stated. Autosign no, and then autosign no for the next one. Read group identifier ID is 000. Sorry about that. 000. Read group sample name, or the SM. In this case, it's father. Copy paste father, and yeah. In general, you're free to choose ID or SM values of your liking, but ID should unambiguously ambiguously identify the sequence run that produces the reads, or at the very least, nodes input datasets in a given analysis should define the same ID twice, or tools like FreeBayes, which are going to be used in the next step, will refuse to work with the data. So more or less what they're saying here is there should be unique identifiers so that the tools will be used in the next steps and moving forward do not get confused. That's the most basic thing you need to know. So yeah, we're gonna run that tool now and repeat the same steps for the mother and for the child, obviously with their own unique identifiers. And everything else should stay the same more or less. Okay, now that we have the reads mapped, move on to the next step of filtering reads. So it's going to explain to you over here more or less that there's different methods to filtering the reads and whatnot depending on the variant calling method you want to use later on. In this case, we'll be using the FreeBayes variant caller. And specifically, before we use the FreeBayes variant caller, we want to only keep the reads that have been mapped and get rid of duplicates. We're going to scroll down here and it tells us more or less what we're looking for, but don't panic if this does not look like what you see when you're working. For some reason that side is outdated. But when you click on the hat and you click on filter, SAM tools or SAM view, yeah, and then we get the steps from here where it says all three mapped outputs. We have those selected. Next, what would you like to look at? A filtered / subsampled selection of reads. Filtered, subsampled. Next, configure filters. Read is unmapped or mate is unmapped. Okay, so configure filters. Configure filters excluding reads of the following flag sets. Excluding reads of the following flag sets. Specifically, any of the following flag set. You want to get any. Because here, exclude reads with all or any. It makes a very big difference. Exclude reads with any of the following flag sets. Read is unmapped or the mate is unmapped. And then that should be good. Read is unmapped, mate is unmapped. Scroll to the bottom. Something else we need to do. And you just run the tool. So the next step after running the SAM tools would be to remove duplicates. You can do that by clicking on here. Okay, let's just stick to this moving forward. So removing duplicates, the three outputs that we got, select multiple, 1, 2, 3, output to the SAM tools. And then BAM is paired end, which we want. Treat as single is no. Run the tool. Shouldn't take too long. Okay, next the duplicate tools complete, and we move on to the variant calling method. Right before you do the variant calling method, I would recommend that you duplicate histories in case you want to use different variant calling methods. In this case, we're going to be using the FreeBayes, but there are things such as deep variant, and you also get your low freak, if not mistaken. There's quite a few options out there. Choose the source, or for the reference genome, locally cached. That is perfectly fine. Run in batch mode. Yes, we want to merge outputs of the VCFs. So merge outputs. Multiple datasets has been selected for you since you want to merge multiple outputs. And then for reference genome, we're going to use HG19. that we're using throughout the entire process. Okay, limit variance calling to a set region. Do not limit. We do not want to limit it. And for choose parameters, selection level, simple diploid calling. And that should be enough to run the tool. Before we move on to the next step, while everything's still loading, you do need to do a careful analysis of your different variant calling methods, as they each come with their own pros and cons. In the case of the FreeBayes method, FreeBayes is a Bayesian genetic variance detector designed to find small polymorphisms, specifically SNPs, indels, MNMPs, and complex events, blah blah. It's suited for shorter or smaller read lengths. Okay, so the FreeBayes, variant calling method is now complete. I can move on to the next step. Okay, FreeBayes complete, get the output. Here's a small little hands-on assessment, just to check out quickly. It says click on the eye of the VCF file, click on the eye, and it more or less asks us, what can we understand from the file in the question? Can you locate at least some of the above mentioned, the above listed information in the data sets? And then it tells you hash means it's a comment, and then you can see homozygous, heterozygous, homozygous to 0, 1. First position of each variant with respect to reference genome is the first two columns. So you get the chromosome and then your position. So hash, there's comments. And here we are, chromosome, position. Next we get columns 4 and 5. So it's the sequence change associated with the variant. Columns 4 and 5, ref alt. So reference, it's supposed to be a T, alternate, it's a G. And get more and more data that you can skim through if you have more time for that. And post-processing of the FreeBayes calls. Before we start analyzing the detected variants, we need to post-process the VCF datasets, just to make sure that FreeBayes, the FreeBayes output, I should say, from variant calling method, and the downstream analysis tools are compatible with each other and we don't run into any issues moving forward. That's more or less what we're doing next with the BCF tools norm. So here we take the FreeBayes output, and then, FreeBayes outputs. Reference genome is built-in genome. We're sticking to the HG19 as stated before. Next, when any ref allele does not match the reference genome base, ignore the problem. Okay. Atomize, no. Left align and normalize indels, yes. Reason being, this is standard practice with most, data sets out there. So please do left align and normalize the indels, just so you run into as few compatibility issues as possible. Next, after that, do not duplicate, and then multi-helical split. Multi-helical, click split option, and then the answer is both, so both SNPs and indels, and output should be an uncompressed VCF, not a compressed BCF. Please make sure to check that. Run that tool, might take a minute to run, but once that's done, we're almost of variant annotation and reporting after the BCF tools. So we need to prioritize filter, and it's reported. Okay, so as you read through prioritized, you can see that we're looking for the relevant genotypes. And for filtering, we're looking for inheritance patterns that match what's going on. And reported is human-friendly form, more or less. Okay, so let's do that step by step. First step would be to get the SnpEff functional tool, blah, blah. We can download it if it's not available to you, but if it is available to you, you can skip that step, not this entire section, that particular step. Because the next step is create a PED formatted pedigree database describing our single family sample trio. And this is a very important step that you should not skip. I made that mistake myself the first time I tried to run this, first couple of times. It's a very important file that we'll need. You can go to this link over here, which will be linked below for the training material, and we'll look for the pedigree.txt. 68 bytes. It's a very small file, but without it, you can't really do much towards the end of this project. Keep moving on to the next step. We'll use the SnpEff. Okay, so we'll run through it step by step. Okay, variance annotation reporting. Okay, so we have the tool open here. So we're going to select the output of the BCF norms. It's already selected for us. Next is input is VCF. Output format VCF only if input is VCF. That's already done for us. Genome source, locally installed reference genome, yes. And genome itself, make sure it's the HG19. HG19, always have to double check that. Can skip the rest of that. And the final thing we need to confirm is the produced summary stats, which is all at the bottom. It's selected yes for us. Don't need to edit anything else. And we can run the tools just like that. Okay, the SnpEff tool is done running. And we can move on to the next step. 2 data sets are produced. Hands-on inspect. Click on the eye of the HTML. So I have the HTML, produces a summary of all the stats / data. Unfortunately, not all the images that should be showing are showing. Yeah, I've ran this several times. I don't think this is an error on our side. Whatever's going on with Galaxy, it's Galaxy's problem. Next, the Gemini load tool. Okay. Can open up the Gemini load. Clicking on it. So we're gonna work with the output. There it is, the output of the SnpEff. There we go. Variants are annotated with SnpEff. Yes, annotated. And inputs come with genotypes called for the sample. Yes, yes. Okay, sample was called by FreeBayes for us. Choose genome annotation source. Choose genome annotation source. There's only one snapshot. Yeah, there's only one snapshot. Sample and family information in PED format. The pedigree file prepared above. Yes. Remember that really important file I told you not to skip that you're gonna need. Yeah, you kind of need it now. So you can always upload data over here, or while you're working with the tools, you can actually click browse, upload, choose local file, pedigree.txt. You upload it, press start. It's almost done instantly. And when that's done loading, you're gonna select that, and yeah, that'll be the pedigree referring to. Reason being, the pedigree database or dataset, sorry, is optional, but several Gemini tools require the relationship between samples, i.e. the family structure, and or the sample phenotype to be defined. The PED or PED format is a simple tabular format. If you choose to not provide sample information now, but later find that you need it for analysis, you can also add it to an existing Gemini database by using Gemini amend tool. Basically meaning it's not entirely a train smash if you don't add it now, but rather just do it, since most of the Gemini tools you're going to use later on require it. Just add it now instead of giving yourself problems to worry about later on. It's literally 68 bytes, it's less than a kilobyte. The GRP score, let's just add that actually. Now that's done loading. We want the GRP score. Yes, you want the CAD score. Deselect the likelihoods, because we don't need the likelihood, and you got only variates pass all filters. Everything else is selected, and then run tool. Once you do that, Gemini load should complete, and we're on to the final step of this analysis. So over here, we're more or less going through the different options as to what we think could have occurred, and they give you the answer if you're willing to go through it. I've been through it before, you can do that in your own time. Moving on to the Gemini inheritance tool. So the answer before was a recessive autosomal inheritance pattern. Okay, so step one, Gemini database. We're gonna take the results of Gemini load, and then autosomal recessive, autosomal recessive. You want to insert additional constraints. Impact severity low, you can copy and paste this directly right over there. Just make sure that everything is written as is. And then include hits with less convincing inheritance pattern. Say no, and then no. Family-wise criteria, we keep to default settings. Reason being, the family is too small for us to really get any valuable information out of editing these settings, so we kind of skip that section. And we go to outputs. Outputs, we want a custom output. Okay, so we go there, click custom, and we want alternative allele frequency. Additional columns have to be included. So additional columns, comma separated, copy paste, exactly we saw there. And then we should be able to run the tool. And once that tool has ran, we should get a result of the different variants that Gemini suspects. And once you've done that, as you can see, we'll reach the conclusion, and you would have finished this tutorial. Shout out to you. You have now completed the, what's it called again? The exome sequencing data analysis for diagnosis, for diagnosing a genetic disease. Thank you for following along. All the best, new projects.